Sierra Nevada field campus. We have the director with us. I don't see JR right now on my screen, but I know he's here and he's at the field campus and he's going to talk to us at the end. So I was up in Sierra City for a couple of weeks. I just got back this week and I took a few pictures, so I'll show you what's going on at the field campus right now after I do my little spiel here, after we get our housekeeping out of the way. A few things. You might want to put your screen on speaker view once Brian and Tanya are doing their presentations, because they have PowerPoints and videos and stuff for you, so that'll fill your screen that way. There's a poll at the bottom of your screen that has 4 questions on it, if you feel like answering that at the end of the presentation. And okay, I got distracted because Hope has a bird on her head. Okay, now we also have a chat box, and you can put questions for either Brian or Tanya in there, or questions about the field campus. And while they're give you my little slideshow, I do want to do some, since this is our last Zoom, I want to say a few thank yous to people. And I was hoping that the Dean of the College of Science and Engineering at San Francisco State was going to be here, but she was at the field campus and she's traveling home now, so she'll get to watch it later. But I want to extend a big thanks to Dean Carmen Domingo. She's the dean of the largest college at San Francisco State, and she really makes the field campus a priority. And she comes with her family every summer and for years, and really has done a lot to support us. And then when friends came around, you know, she's been really working closely with us. The other person is Jason Porth, who's the head of the University Corporation at San Francisco State, and he also comes up to the field campus every summer and is just a great supporter, and in the last year has committed a significant amount of resources to improve the infrastructure on campus. And so he's really accessible to us and really cares about the field campus. And I just want to say thank you, Jason. It really means a lot to us, all the students and all the friends that we have. And JR's been director for 11 years, and so we should really acknowledge him for hanging in there with all the things that come flying his way every summer. And Kristen's clapping, yay, JR, hang in there. It's been a lonely summer, but we'll get to show everybody some of the things you've been doing. And then lastly, our presentation today is with 2 classes that are made possible at the field campus through the College of Extended Learning. We call it CEL. And the CEL classes are for credit. So people can come and get college credit for any of these classes, 1-week-long classes. And this year, CEL, which has kind of a complicated registration process, went out of their way to accommodate the field campus pre-pandemic, of course. They changed their registration process for us. They worked with us. They paid for the printing and design and mailing of our schedule, which was huge. So big shout out and thanks to Dean Alex Hugh of the College of Extended Learning. Okay, so I think that's it. And then I just want to share a few pictures. I just have 10 pictures of what's happening on campus, all the repairs that are going forward, and a couple of other things this summer. So I'll just get that started. And so friends purchased a portable classroom. As our enrollment increased, we really needed more classroom space. And this is a 14-by-16 canvas tent. Kind of get an idea of the scale there with JR. It's a really nice tent. It's going to work out really well for us as a classroom, extra classroom. The outside showers have gotten stripped and water sealed. The wood is protected more. And here's the staff cabin. Looks kind of crummy now, although you get to see that nice new roof that the Friends of the Sierra Nevada Field Campus put on last year. So that the brown painted part is what the whole thing is going to look like. It's probably a lot further along because this was a couple weeks ago. And here we have the women's bathroom. Am I hearing applause? We got the nice new doors on it, and you can see the ceiling is now finished out. And then in the shower section, the water heater is in a closet now, and the cubbies are all finished out. So here's the men's, and it's going to look a lot like the women's. They'll have a nice place to store all their stuff there next to the showers. And then we put a new wall in, and that's where the sink and the mirror will go. And the toilet stalls will be just like in the women's. So the tent platforms have gotten water sealed. It's going to make them last a lot longer. And here's just a view for you of the dining hall and kind of what our lonely field campus looks like right now. It was nice to see that the lupin was just carpeted all over the outside the buildings. And yeah, the North Yuba is super low right now. If you've been there, you recognize that scene. Okay, let's keep our fingers crossed for 2021. So, yeah. Okay, well, let me introduce our Tanya Baxter, who is a botanist, and she teaches 2 classes at the field campus, the Flora of the Northern Sierra Nevada, and a new class we were going to have this year, which is called Trees and Shrubs. And so she's got a presentation first, and then Brian, I'll have you just take over after she, so I can get out of the way. Brian is, Dr. Brian Perry is a mycologist and head of the biology department at CSU East Bay. And lots of you are here, probably know both of them already. But if not, you'll get a little introduction to what they do at the field campus. And I don't know, is there have I forgotten anything, Tanya or Brian? Yeah, that's good. So we'll bow out, put your mode on speaker view, and I'm gonna say goodbye until I come back at the end.
Okay, can everyone see this really clearly? How are we doing? Good? Okay. I'm seeing some thumbs up. That's great. So I want to welcome you to Flora of the Sierra Nevada abbreviated version. This topic tonight, we're going to focus on, did I get this scrolling first? You're up, great. So we've had some interesting weather, as you all know, and I think a little bit of it reminded me as if we're actually in the Sierras right now. So if I'm going to invite you to have sort of an experience as if you were there right now. So I invite you to just think about that fresh smell of the rain happening, and it's nice and hot, and the thunderclouds have already passed, and it's just post-storm, and you're now sitting in a meadow. Maybe you're at the meadow overlooking the Sierra Buttes. I don't know, wherever you shall think. Just go ahead, close your eyes for a minute, take a second, visualize yourself there, and then when you're ready, go ahead and open your eyes again and follow along as we take ourselves there.
Hello.
Oh.
I'm also recording my voice.
So one of the wonderful things that we have here in the Sierra Nevadas is like a wonderful demonstration of really showy corolla, showy flowers, and plants that are from a historical time. in a lot of California, we haven't had a chance to see things in their natural state, undisturbed. Really, we have to think about that on the global sense. You think of Europe or any other places that have some sort of Sierra montane landscape. We're really fortunate to have this sort of setting in which we can see things in their true natural habitat. And so as we go along, we're going to notice a lot of patterns in our plants. We're going to kind of want to look a little bit more closely and ask some questions. And where do we begin is a big question that I get, because I can look at leaves, I could look at the flower parts, I could look at the fruit, you see it in bud. It can be very confusing. But today we're going to just focus on one group, and we're going to try to pick up some patterns. And I feel like, since I don't have specimens in hand and we're not on the field campus, we're going to really dive into this one called the Ranunculaceae. As you can see, we have some fine specimens that maybe some of you are familiar with already, hopefully have seen yourself. This is a delphinium, and it's a tribe of the Ranunculaceae, and it's a Delphiniae, has 2 different genera, and it has about 9 or 8 species that we generally see within the delphiniums. And there's a distinction here that I'm kind of focusing on quite a bit. As we get closer, it's going to be in the way the display of both the stamens and the pistils. Even though we are very much noticing the corollas on these structures, I want us to kind of take a further in look, because a lot of these characteristics that we focus on, this is a good example right here. This is a little bit of a hole visiting by a pollinator. Tends to be, superseded by nature. They're after something more, and it may not necessarily be what we're seeing typically. That's the corolla, right? So evolution has actually driven us in a certain way with these pollinators, so that we get to see more of these nectars that they're being attracted to. Bulbifery is also another adaptation that we see out in the landscape. But the corolla, the showy parts, that's what we end up finding ourselves focusing on. So I'm gonna take us on to the next slide here. So I just wanted to give you a quick overview of the Flora, the Sierra Nevada field course. We spent about a week exploring the region of the Great Lakes Basin. Is rather untapped. It's volcanic and glacier primarily, with a little bit of quartz dikes from some uplift over the years, and that has led to some mining. And our course is going to focus a little bit more on spending time in the classroom, and then in the afternoons we're going to get some hikes in to go see these things in the landscape. And we have opportunities to do some keying sessions in the afternoon, and that tends to be really popular. It just gives us an opportunity to practice. And keying is going to be using some of the taxonomic books that we have available for us. And by then you'll have been a little bit more familiar with some of the terminology. I'm going to be throwing a lot of terminology in this course at you. However, for today's session, I'm purely just going to focus on some of the basic parts of a plant, so that we can walk forward with that. And then also I'm teaching a course that's going to be on trees and shrubs. So this course is going to go on probably towards the end of the same week. And in case you want to just get a gestalt and become a little bit more familiar with just the larger landscape, we can start there. But the Flora is rather content heavy, I'll just warn you. So let's see if we have time. This is going to be, by popular demand, maybe a little bit more of a video of what we find on the field campus and what your experience has been like. Classic Sierra Buttes, getting an opportunity to see some of these plants up and close. And this is a penstemon. We have a Saxifragaceae family, which might be a new addition next year. Visiting some new places like Babbitt Peak. You guys may recognize the director. And this is some of the activities we'll be doing is looking at plants through dissecting scopes and also with hand lenses. The hikes are within the vicinity of the field campus, so we don't have to go very far, fortunately. And we visit numerous plant community types, including this bog that is in the Feather River Canyon and produces some stunning arrays that we will not necessarily get to see anywhere else, of some plants that you are probably familiar with these Darlingtonias. Keying can be done in this field. That's a great way to get familiar. And we may have some time to go for some dips in the creek, which if we're lucky, that might be nice. Higher elevation, we're gonna get to go and see some of the rocky scree habitat. And there are numerous plants that are there later in the season, so you can always go back and see some of the representations after the class. And if we are lucky, we'll get up to the buttes themselves, which is a nice treat. So there we go. So just to give you an idea of some of the tools that you're going to want to have on the course, this is hopefully, if you're looking up at my screen, but the top right corner, this is a hand lens. It's a 10X, pretty small. I'm going to try to bring it up to the screen. I'm not working with this great technology that Brian's working with today, so I'm going to do the best I can. These come in a loop. I highly recommend the loop. This was Dr. Bob Patterson's big emphasis because you don't want to lose one of these. And you can wear them. I will show you how. And then also we want to do a 10 centimeter ruler. Pretty simple. Sometimes you can clip them and you can get them attached to your hand lens if you like. A set of tweezers. You can get these anywhere at a drugstore. And then I carry around with me is a little pocket knife for cutting. So it can be handy, but it's not required. Other tools that you're going to want to have, if you happen to have it, this is the book we're going to be working with. This is called the Jepson Manual. Hopefully some of you have seen it before. It's a rather large book. And so you can get a lot of this information online on the Flora. Mine just happens to have a carrier in case. Yours doesn't necessarily have to, but it can be handy. And the Flora has been a tremendous help. But however, in this course, we're not going to actually be emphasizing too much on using internet sources. So this is a field course, and I think that's actually fantastic in my opinion, because we can get a lot more out of it from seeing things out there in the field. So Jepson Manual, if you have a chance, please be sure to review the glossary beforehand. It's a great way to familiarize yourself with a lot of the terminology that we go over. And there's some wonderful illustrations in there as well. Lastly, if you want, plant presses are highly recommended if you want to take them back with you. Mine was homemade built. I happen to really love this one. And they just come with a little bit of blotting paper in between and some cardboard, some newspaper to hold onto your specimens so that you can look back at them, and we want to make sure you have a good record. So we will go over that, how to collect a plant. That'll be further on in the course. I'm not going to cover it too much today though. And then also, how could I forget, California Native Plant Society, the chapter here has been wonderful at creating a book. This is a print. This has actual images. It's going to be around. I wouldn't say that we're going to be spending too much time using it, but it's very handy for you as well. And Bill Wilson was involved. He's one of our editors, so I got to put a big shout out for him on that. So thank you. So there's a lot of plant families in the Sierra Nevada that we could cover. And for the course, I only focus on 8. Today, we're going to do 1. There's 4 of them that are typically above in this, I should say the alpine zone, maybe about 9,000 feet and above. We're about 8,500 generally in the Sierra Buttes area. So the Asteraceae, the Brassicas, the Cyperaceae, and the Poaceae cover about half of the alpine flora in general. So that's kind of exciting if you ever want to just focus on that region. But majority of these actually extend their range into the foothills. And so sometimes we will actually be working lower in elevation depending upon the season. Last year was a good example of that because of the late snow melt. So we're fortunate in that sense. So I'm not going to go into these. I'll spare you the moment. So some plant parts. Well, this one was kind of easy. I decided that, well, why don't we start simple, right? Does everybody know what a receptacle and the sepal and the stamens are? Over here, this is, can anyone guess? The stamens are the male part. This is going to hold the pollen. The pistil is the female. And below is going to be the ovary and the style. The sepal and the petals, they separate out. The petal is the structure that we often see that's the most colorful, and it's going to be adjacent to the stamen. Below that is the sepal. And then the structure right in here, called the receptacle, I emphasize on it because that can actually modify quite tremendously as it starts to go into fruit. So here we go. Hopefully we all know these by now. Let me know if you don't. I'm going to be using these terms interchangeably, but a real quick overview. Here we go. Stamen, right, as the structure as a whole is the male structure. This is the filament. This is the tube that holds it, and then the anthers that actually has the pollen. And then we have over here the carpel, or also collectively known as the pistil, typically. And then the stigma, which is the receptive part, the style, as well as the tube, and then the ovary down here. Now the interesting part is that there's a lot of these, the ovary can vary quite a bit. And there's chambers, and there's different shapes, and if there's a fusion. So oftentimes when we're keying plants, this is where the emphasis is, and that's the odd thing, is that a lot of times when we go out as botanists and we're just seeing things from afar, we have to actually get very close. We have to get further in to just start to distinguish things. And so that's something that we want to kind of bypass the showy part of the petal and start to notice these patterns. So that's what'll be an emphasis when we get to looking under scopes and we talk about distinguishing the plant families as we go forward. The sepal, it doesn't necessarily always have to be photosynthetic and green. It can actually have some pigmentation as well, and have some, and a collection of them can actually be fused. And the ovule, of course, being the smaller of the chamber of the ovary, where the seed develops, excuse me. So here we are, we're gonna dive right in. Why did I choose Ranunculaceae is what probably people are wondering. One of them is it's a rather ancestral plant, it's primitive. I always loved magnolias and thought that they were really stunning, and was really excited to learn that actually the reason that they're, you know, these big fleshy petals and you know, this strange collection of the stamen is because it's such a primitive plant. It's just has like the parts are just really kind of, they're very simple, all of them. And then yet, you know, in the Ranunculaceae, which is closely related to Barbaridaceae and Papaveraceae, which is the Papaveraceae is our poppy, the California poppy. And the reason I find them fascinating is that we started talking a little bit about the display of some of the simpler parts. So even though they're ancestral, like the carpels, how they represent themselves, the numerous pistils can be a very simple in a lot of ways, because it's not a fusion of them or something more elaborate. Say like if we cut open a tomato and we see numerous chambers, that would be something that's more, as what we would call derived, evolutionarily is more advanced. So there's some characteristics that are retained, and then there's some that are acquired as time has passed. And this is some of our oldest plants, which is kind of exciting to think about. And look at how fascinating they are. It's really kind of cool. Okay, so we'll come back to that slide. Don't worry, that one won't disappear. So there's some terms, here we go. So the two things I focus on, the derived trait being something that's a little bit more nuanced. The ancestral traits are these, where the petals, there isn't necessarily a pattern yet. So something we often will see as plants have evolved is that we distinguish some things that are, we call them meris as a terminology. So if you see things in multiples of 3s, so you see 3, 6, 9s, 12s, then we pick up those patterns really quickly. And as you're probably familiar with your lilies and orchids, that's a distinction of the monocotyledons. Ranunculaceae, there isn't really a pattern. So we could have maybe no petals, or maybe we have several petals. And a poppy is another good example of where we see that. There isn't really, we can't count the number of petals that we see in there. Similarly, the number of stamen they're numerous. And also the pistil is rather simple, is one to many. So the ovary chamber is rather simple as well. And I'm going to show you some examples of that, because that sounds really confusing until we get to see some visuals. The seeds could be aggregated, they could be in a cluster. And then the fruit as well is rather simple. These are distinction. This will separate them out from other families. So it's a collection of characteristics that are going to allow us to better understand them. And then also the beak on the style, on the fruit is retained. So the style retains on the fruit, but it can be curved, and it can show up elongated as it develops. It could have hairs all over it for wind dispersal. So that's kind of an interesting trait that's kind of moved on. And also the leaves. So the great thing about Ranunculaceae that I also enjoy is that even though we may see it in the spring and we see it with the corolla, you can actually see the plant if it's with leaves and fruit. And I feel like you can be confident enough to be able to identify the family. So if I'll say that again, so you can see just the leaves and you can see the fruit, and you should be able to ID the family. And we're only working with about 8 genera, so it's a good place to start. So here we go. Which ones are we going to start with? We have quite a bit. The ranunculus, as you can start to see, has this. Are you starting to pick up some patterns already? The numerous stamen, those are the first ones that you can see right away. We can't necessarily see the pistil unless we look at the caltha. And then also over here, the thalictrum, there's 2 photos because there's actually a female and a male plant. They're very separate. All the flowers are actually on separate individuals co-occurring in a population. Clematis is another interesting plant. This is one that has that style, the style being the structure above on the female organ, if you remember. This is the ovary, and then the tube, and then at the end is the pistil. But interestingly enough, as you see it in flower right here, you don't really notice anything about the pistils quite yet. But as they start to develop, they elongate. Enough so that they're so wind dispersed that they have like a little bit, there are feathered hairs on them, and so they can spread easily. So it's kind of a fascinating feature, and that would be considered a derived trait. So moving on to leaves, I think I mentioned the leaves. So deeply lobed or divided, and there's some patterns that you can start to see right away. They're also pinnate. Pinnate meaning that there's some branching going on. And here's a good example of it right here. This is a main stem. This may actually all collectively be called a leaf. And this may be off the side a leaflet. I don't know if you guys can see my stylus, but hopefully you can. I'm pointing to the contium. And then this lobing as well is distinctive. This one has some teeth marks on it. And these characteristics will be really important as we get further into the key. We're going to be starting there'll be some questions that'll be asked of what that looks like. Okay, so all right, we might come back to this one too because I find this one really kind of interesting. Okay, so we want to learn some new things. I thought I'd start with something, one of the most well-known and also probably one of the most well-studied is the Aguilegia formosa. This is the Western columbine. Columbine has a large geographic range throughout the world, and oddly enough, it actually has been, the genus itself has been evolving at the same time, simultaneously at the similar rates as other Aguilegia throughout the world. And some of these drivers that have been pushing it forward in terms is maybe pollinators. Pollinators syndrome on this one, as you can see, is rather elaborate. You can see this sort of long structured spur that has been created, probably because of a push towards, say, hummingbirds and also butterflies. But a number of pollinators are going to be driving that in a given location. So it's very specialized. And they're typically going for the reward, which is the nectar of the tube. And so as they're inserting their proboscis inside this tube, they're actually going to be brushing up against the stamen where all the pollen is, and then at the same time serving as a mechanism for pollination. So another thing that we see is this geographic isolation that's kind of been a driver for some flower characteristics in other parts of Europe and Asia, which makes a lot of people love to go and chase down Aguilesias all over the world when you travel. So if you're thinking of a new, hobby that you want to take on in retirement, this actually sounds kind of fascinating because you get to go to some very scenic locations. And I don't know, I can give you some tips if you want to know a little bit more about where to go. So anyways, there's a lot of hybrids that we now see in the horticultural industry that have popped up out of this. And also there's just sort of like you get a sense of this continual evolution pattern even in our area. And I think I pointed out in the slides is the video that there was a little bit about here. Let's see if my stylus shows it. There was a hole right here on one of the, on the corolla. And that was probably because there was an insect that came in and just decided to take a shortcut to get to the nectar. And they just decided to just munch away right here. So number of people will start to study this sort of pattern to see if this is actually going to push plants to have some adaptations in different ways, if that continues to persist, right? If that becomes more of an issue or not, you know, these sort of things just pop up in nature in general, because there's not just one or many pollinators that are visiting a plant, but they will often sort of, there'll be some things that you can pick up, and I'll describe a little bit more about that later. But this particular genus is very old, 6.5 million years old, and it was about 3 million years ago where we started to see this branching off. So I encourage you to kind of fall in love with Aguilesias if you can't now, if you haven't already. Another one that's rather widespread in North America, the Acontium. This is a monk's hood. This flower, this is from a research article, I have to say, that I really appreciate how she was able to capture it, because I don't have any specimens. I would open one up right here and show you how it works, but it's a really neat adaptation. With it, they have these nectary glands up here at the very top. There's a pair of them. And this, where I was talking a little bit about how there's a little bit of a evolutionary push going on with bulbifery. In particular, since it's such a large range, this is pretty much all of North America, if you will, right? And the continental 48 states that this spans. However, in the Cascade region, there's been some studies that are looking at the amount of bulbs that start to develop is actually correlated to there being a little bit shorter depth of the nectar. So maybe there's some push in terms of geography in these sort of locations that there might be better off adapting to spreading by bulbs. So these are kind of fun plants, and as you can see, the nectar spur length is correlated with bumblebee tongue. It actually gets visited by a number of different pollinators. So it just happens to be that has been found in this particular subpopulation. So kind of exciting. See where it goes. Another one, Phylloxera flendleri. So one thing I didn't really mention about Ranunculaceae, there is actually, it's got a little bit of slight toxicity to it. It's nothing that I think you can get dermatitis and if you decide to ingest it, then we might want to have an issue, but I'm not planning on anyone having a salad out there with this, so fortunately. Phylloxera flendleri has these sort of like adaptations, if it looks like it's rather wind pollinated, if you will, and that is because there are two separate individual plants that have all flowers on that one individual will be all male and the other one will be all female. And we call that dioecious. So typically we don't see that. Typically we see all of those structures within one flower. That's maybe about 70% of the flowers are that way. But this dioecious pattern has evolved in our Phylloxera. And it's rather showy, kind of distinctive. It looks like earrings. They're going to be found right there on the field campus. I'm sure you've walked down right to the creek and seen them yourself. And I appreciate that they have those leaf patterns that will give it away. That is the clue, the clincher on this one. So fruit chambers. So we're going to go into a little bit about the fruit. The article. Ooh, I'm noticing how hard this can read. Hopefully you can all read it. It's a little. These are the illustrations that come from the manual, and they were probably about that big or something when they started. The follicle that we see over here on the left, I believe there's so Columbine has a great example of this. We're seeing a little bit of the example of that beak pattern that comes on. This is maybe a derivative of the actual seed itself, or also the style. As well over here, let me see, I gotta move my cursor a little bit. On the keen, this is another beak. That is kind of curved a little bit more. So it's not just this residual style that's gonna fall off. It may have actually started to curve a little bit. So that's a kind of neat characteristic we see in the article as well. And so the difference between those is a little bit of the ratio of the size of the seed to the entire endocarp. So let's keep an eye out for those ones. Let me see if I move my cursor there, sorry. And then the the partitioning that we talked about, if you take a look at a good example would be, let's focus on the follicle here, because I think these capsules are something more you would see in other plants. The follicle, if you notice, it's separate. This is a little bit more of a fusion. This would be something like your cucumber or your tomato that has the chambers that are starting to fuse. The follicle, each of them are still separate. And this is one of the derived characteristics. This is one of those characteristics. When I say derived, it means older. It's retained with evolution. So we'll see more patterns of that with the Ranunculaceae. Okay, so the achenes as well the urticle, all separate. Okay, let's keep going to the next one. Here we go. Okay, this may be a little bit better slide. Here we go. Let's see the real stuff. So there are berries. This is a baneberry actually. And there is the beak. You can see it right there. And here's a nice example of our clematis with those feathery, elongated styles that blow in the wind. And many of you know this is columbine. Many gardeners will collect its seed because it's so easy to. See how it dehisces? It just opens up, separate. All right, so I talked already a little bit about clematis. I jumped ahead on that one, but we can also just appreciate it as a wonderful vine that is in the Ranunculaceae family. So we mostly see herbaceous, perennials. This one happens to be a nice vine. Okay, so the inflorescence shape. So now we're moving more into the derived categories. And so this is a characteristic where we see a divide in half, common in our delphiniums. And if we dissected a monk's hood, it's as if we took a book and we could open it back and forth. But if it was a pie, we would have 8. We'd have, or 6. If we could cut it in different parts, it would be the same altogether. That would be a radial characteristic. And we also want to be looking at the way in which the flowers start to develop as they unfurl and when they're young. Sometimes the younger plants or the younger flowers and bud will open closer to the stem, or they'll be at the very end, which is terminate. So this sort of indeterminate determinate can be a real factor when we get closer to the taxonomic keys. And so we'll not focus on that too much today. We're not going to overwhelm you, like I said. So going back to some of the flower parts that we wanted to focus on, this is a great example of cutting it in half, the 2-book version, and this is the radial. So we have both examples, as well as the aguilegia as well. Zygomorphic meaning that there's also some fusions. So delphinium happens to be one of our larger groups. I say the Delphiniae tribe. Believe it or not, there are more tribes, which means they break down within the Ranunculaceae. I'm not going to make you learn all those, but it's a nice way of clustering them and getting to know how they're associated. And the Nuttulatum and also the Andersonii are 2 that, oddly enough, even though they exist in very similar ranges, they tend to hybridize, and their fruit can be a distinction. So it can be a little bit challenging working with the delphinium keys and at times. That said, I think that you'll notice right away that separates them from the other delphiniums is going to be the type of leaves, actually, oddly enough. It's these deeply lobed leaves. So rather than the other delphiniums are going to be a little bit more shallowly lobed and large and like maybe more palmate, they're going to be larger in stature, maybe a couple of centimeters in size difference. So those are some characteristics. I would really like to actually divide, if I could dissect this a little bit more to show you what the pistil looks like inside the delphinium, but I don't have it right now in front of me, but you can sort of see what it looks like over here. It's tucked back in there, but we could show you a little bit more about, I wanted to focus on these sort of ancestral traits that distinguish the delphiniums in this case. So that's a next time. when I have fresh material. So another great genus is the ranunculus. Did I say genera of the ranunculus? Yeah, the ranunculus genus as a group is can be easily mistaken for the Asteraceae. So if we all know the composites, they typically have those beautiful yellow ray flowers. But the thing is with the asters, each of those flowers is a separate flower on an individual head. So we just have to remember what I said. If you have fruits and you have leaves, you can drive at home with Ranunculaceae. And so I'm going to bring you to this image right here. If you take a look, you can start to see that little style beak coming up off of each of these achenes. This is the fruit, the immature fruit that are starting to develop. And that'll give it away. So each of those is kind of separating out. As you can see, they look kind of like they're dividing out in all different directions. So that's the very simple characteristic. And then that style beak at the tip. I haven't done a time check, guys, sorry. I'm going long. And then we also have a couple of others here that you'll get a chance to see, the Actea rubra, which is a little bit larger red fruit, and the caltha. There's the ranunculus, we can find a number of them in wet meadows. So we have a lot of taxa of those. Okay, so I'm wrapping it up here, so hopefully I'm not dragging you guys too long. And so lastly, I wanted to give you an opportunity to know a little bit more about how it works with what we do in herbarias. Herbariums are what people are doing to keep all these plants records and stored and mounted in specimens. Oftentimes when we're collecting, we may bring also little envelopes to collect seed when we're putting everything into a plant press. Plant presses, remember the plant press. You're actually likely going to be digging the whole plant out if you're going to be doing this properly, so that you have some examples of the root matter. You want to make sure you have the bulbs, or maybe it's a tuber, it could be lots of other parts. So you want to be sure that that's collected, and it's good to collect more than one. Of course, there's a rule on collecting in terms of how much you come across, but you want to have a handful. And then there's some that are just not going to make it really very fast, so you have to make sure that they get pressed right away. Like our Clarkias, they lose their color pigmentation super fast. So proper mounting is a key, and then you can do these wonderful illustrations that hopefully maybe Kristen or someone else can lead afterwards and show us how to do. But I too have a dissecting. This is what we would use as well. I have one, Brian, I promise. But the dissecting scope, we will use these heavily in our class. And this will give us a chance to look at some of those parts that we can't necessarily see right up close, like the pistils or the fruit, and dissect and cut them in half and get in there. And then we'll also really get to appreciate some finer characteristics, like the hair patterns on some of the plants are really quite stunning when you get to see them under a scope. And it's kind of magnificent. So. Okay, so next year I'm going to introduce some new plant. This is a taxonomic key. I didn't send you guys through one. I was kind enough not to. But do you get a sense? This one's actually a nice one because it's really straightforward to start with. But Saxifragaceae happened to be one of my first loves for plant families, so I think I'm going to bring it back. And it's a great one for the Lakes Basin area because it's just like as well one of these older plant families, and they're delicate and they're subtle, and so we'll have some fun with those. So I think that was the last I have of my my presentation. Hopefully you guys all feel really comfortable going out there and getting to know the Ranunculaceae. And if you have any questions, feel free to reach out to me. I'm also going to be doing, like I said, the Trees and Shrubs course. That's going to be much more of an abbreviated workshop for the weekend warrior type. Or it also can be an add-on to the course that we're doing. I just decided to separate it out. I normally included it, but by popular demand, we just are going to not overwhelm you all. So, all right, with all that, I want to thank you so much for coming out and hearing a little bit about plants. And I want to introduce to you Brian Perry, who's a mycologist and has been doing some wonderful stuff in the East Bay as well as up in the Sierra Nevada Field Campus. And so I'm going to hand it over to you, Brian.
All right, thanks, Tanya. That was really great. Yeah. Awesome. Now I'm going to come take your class.
Great.
Sure. All right, so let me share my screen here. Get to my PowerPoint. All right, hopefully everyone can see that. See my screen. Okay, so as Mary mentioned, I'm the teacher of the spring fungi this here in Nevada, and that's Biology 315. So a lot of folks do come up and take the course for credit. We get a lot of folks from the Forest Service and other state or federal agencies that are doing essentially a lot of their continuing ed credits and things will come and take the class. So we get a big diversity of folks that come and take it. And the class has been running for quite a number of years. Last year was the 35th offering of the class. This year would have been the 36th. So it's been going for more than 35 years because we've had a handful of years where we had to cancel due to really severe drought, or one year the instructor wasn't available. But it's been going for a long time, and we've been keeping records of every single year. So we have a really great database of all the fungi that we have found up in the Yuba Pass area for the last 35 plus years. I'm the third sort of permanent instructor of the course. I took over from the previous instructor, who many of you probably know. This is Dr. Dennis Desjardins right in the middle of the photo. He still comes up and hangs out and harasses me. He gets to play the role that I used to play as being the grad student hanging out and drinking his advisor's wine. Now he does that to me. So he still comes up for a few days each year and helps out with the class. So it's a lot of fun. So what my goal today was to sort of give you an idea of what it's like to come up and take the class. And I thought the best way to do that would to start out by, let's just go on a virtual walk through the woods, and you can see what it's like to collect mushrooms. Now, I apologize. I had a beautiful, wonderful narration to this whole video, but Mary and I discovered about 5 minutes before we started that for some reason the sound doesn't come through from Mac to PC. There's some sort of file issue there. So I'm going to do my best to narrate over this, see if I can remember everything that I said. I recorded this back in early June when I would have been teaching the class. I came up to check on the field sites, and I wanted to still record as many species as I could so we wouldn't have a gap in our data. So let's let it roll here and you can see what it's like. So what I'm doing here, I'm just walking around the actual field campus. So I'm down on the lower end of campus walking towards the river. The director's cabin would be off to your right if you're walking through the woods here. And when you're collecting mushrooms in the Sierra Nevada in the spring, it's a little bit different than it is if you were collecting, say, on the coast in California. What you're doing is you're walking around and you're really looking for little lumps of duff that have been pushed up from underneath. So in field guides, they often refer to these as mushrumps or as shrumps. And what's going on is that the fungi are producing their reproductive structures. So mushrooms, or basidiomes as we call them, in the case of the group that we're looking for here. And those are going to fruit several inches underground. And then as they grow and expand, they're going to push the soil up. And this is a really common way for the fungi to fruit in the spring up in the Sierra Nevada. So you're not just going to find these big giant mushrooms standing above the soil. You will find things like that occasionally, but usually you're really just walking around looking for lumps. And then you go over and you see these lumps, and you essentially start to dig around them and see what you got. And so it's kind of hard to tell from the video here, but I can see all sorts of little shrumps all over in the woods here. And so of course I pre-scouted out the site. I knew what was here. And so walking up, I'm sort of approaching one of these shrumps right now, and hopefully you can make it out. And I'll kneel down in a second here and show you. But what I'm looking at here is just the cap of a mushroom that's pushing up through the soil. And so what I'm doing is I'm just very gently sort of removing the dirt from around the edges of it. And in a sec, what you're going to see is I'm going to take out my collecting knife, a pocket knife, and I'm essentially just going to dig underneath that mushroom, and I'm going to try and pop it out of the ground without breaking off the stipe. And I know what species this is already because I've been doing this for a long time, but you'll see in just a moment when it comes out of the soil that this is going to be our Boletus rex veris, which is the spring porcini that we get in the Sierra Nevada. And so this is one of the most highly sought after mushrooms up in the Sierra in the spring because it's a true porcini, right? This is a really good edible mushroom, and it's kind of hard. It's a little bit out of focus here because I'm too close to my camera. But what you can see is that when you look at the underside here, it doesn't have the gills of your normal, white button mushroom, but it has pores instead. And so this is, keep in mind, this is the reproductive structure of this fungus. And so that would be a young button of Boletus rex veris, the spring porcini. Whenever you find one mushroom, you want to look around because you're probably going to find more. And so I'm just sort of walking around this little area, and there's literally shrumps all over the place. And so what I'm showing you here, these again are two really young buttons of the same species, that's Boletus rex veris. And then there's a few more over here. And so notice here as I start to push that back and pull the soil back, you'll see a lot of insects that are buzzing out of there. And I'll say more about that in just a second. But looking at this, if I get wait for the camera to pan down, there we go. This is a huge cluster of what would mature into a really massive fruiting of these porcinis. This would be a really prized find if you were out there collecting for the table. And so these are really young. They really haven't had a chance yet to produce and disperse their spores. So I tend not to pick mushrooms like that. In this case, I'm just going to cover them back over with a little bit of the dirt and duff, just so they don't get dried out by the sun, right? So give them a chance to actually continue to grow, produce their spores, and then disperse those spores into the environment. And again, looking at this one, notice all the flies that are coming out as I dig around these mushrooms. And for those of you that have ever collected porcini, you know that a lot of times when you start cutting into them, you'll find little maggot tunnels. And the reason for that is that these flies are getting in when these fungi are just starting to produce their fruiting bodies, and those flies are laying eggs. And those eggs hatch, and the larvae then burrow down into the mushroom to use it as a food source. So a lot of times when we collect these, we'll find maggots inside of them. Just a little extra protein for you if you're going to eat them. Okay, and so this year, while I was up there, early June was a really, really good year for porcini in the Sierra Nevada. So people were up there collecting tons and tons of these things. This would have been a really great year for the class because the students would have collected a large amount of porcini, which means we probably would have eaten it for dinner on probably every night that we were up there. We would have got the camp chef to prepare them for us, or people could have taken them home and enjoyed them after the class was over. But again, these ones are still really young. So I'm just going to sort of bury them away, let them continue to grow and make spores. And here we have another mushroom, and this is a different species. Okay, so you'll see here when I pop this one out, it's going to look very different underneath. So looking at this one, rather than having pores, this one has gills, or lamellae, as we call them. And I'm smelling it because a lot of the mushrooms up there have really distinctive odors. We get a mushroom that looks a lot like this that smells like raw potatoes. Some people think it smells like cucumbers. It's much more of a raw potato odor. Again, I wasn't sure how close I could focus with my camera. This is Hygrophorus purpurascens, and it's a really common species up there in the spring that we find all over the place. Okay, and so you can just pretty much get an idea of what it's like to collect. I'm just wandering around the woods rather aimlessly, and when you get into an area where there's a lot of mushrooms, that's an indication that there's enough moisture in the soil. In this case, I'm not sure if you can see it, but there's all sorts of red ants all over that mushroom. So I left that one be. I didn't want to get a bunch of ant bites. But these little buttons for the Spring King were just everywhere up here. Okay, so lots of porcini this year, all over the field campus and pretty much all throughout the Sierra Nevada. People were posting lots and lots of photos of collecting these throughout the entire spring season in the Sierra. And then finally, I think this is the end of the video, just another one. You can just see how that mushroom cap has just pushed its way right up through the soil. Okay, so hopefully that gives you an idea of what it's like to collect. So the way we run the class is that we go out in the morning and pretty much I'm going to lecture for an hour or two every morning after breakfast. Then we make our lunches, we head out on a field trip. We'll spend the entire day in the field, eat our lunches in the field. And then around 3 o'clock or so, we'll come back to the laboratory, okay? And I just wanted to show you a few more pictures of other fungi that we find up there. So in addition to things like Boletus rex varus, we find a whole suite of species that are associated with snowbanks. Okay, so most of the moisture that's in the soil up in the Sierra in the spring comes from that melting snow. And there's a whole suite of species that are really closely associated with that. So right along the edges of these snowbanks, you'll find specific species. And so they might be growing just right along where the soil's really, really moist. Or in some cases, like Mycena navicola here, they might even melt their way right through the edge of those snowdrifts. Okay, so we'll spend a lot of our time during the week going up to like Yuba Pass area, driving out Weber Lake Road until we can find snowbanks that we can collect along. All right. We've also got edible species. So I already talked to you about Boletus rex varus, the spring king, but we also have things like Morchella, right? So, or morels, if you know that name. So we have a handful of different morel species that occur up in the Sierra Nevada, and we typically find a good amount of those during the week as well. So a lot of folks do come up because they're very interested in learning, more about edible species, more about foraging. So a lot of folks will really sort of hone in on the edibles during the week class. But I try and get folks to look at everything. Okay. We also have a number of rare species up there, and these rare species may be something that we only see every couple of years, or they may be something that we've only seen once or twice in 35 plus years. Okay, so we have some really, really rare taxa up in the Sierra Nevada. And so for us, whenever we find these, it's really exciting. Also really exciting for the students to, you know, they bring something up and I start jumping up and down and get very excited about it. That's a positive experience for them. Okay, so we get pretty excited when we find these very rare things up there. And then of course, new species. So we've been collecting up here and, you know, running this class for over 35 years now. And part of that documenting of species has been discovering undescribed species. So we have a handful of species that we've described from the field campus and Yuba Pass and that whole area. And this is just a handful of some of the more recent ones that we've described. Okay. So as I said, we'll spend our days in the field, and then around 3 o'clock, we come back, and that's where the sort of the technical side of the class begins. So we take that whole downstairs laboratory, we take that whole thing over, we set up microscope stations such that each student gets their own microscope and their own compound dissecting scope. And we then have a series of guided exercises where what we do is we teach you how to use a microscope for mushroom identification. So a lot of the folks that come up and take the class are really there because, number one, they want to learn how to use maybe their own microscope that they've recently acquired or that they've had forever, and they want to, you know, reacquaint themselves with it. Or they're amateur mycologists that are trying to take their skill set to the next level. Or, as I said before, a lot of folks satisfying continuing ed credits. But at the end of the week, I can guarantee you you'll be a very proficient field collector, and you'll also have a really great skill set at using a microscope for mushroom identification. Okay. And so what I want to do now is I want to sort of take you through, as I said, I want to give you a real taste for what it's like to be up at the field campus doing this. And so what I want to talk about now is using microscopes for mushroom identification. So I said before, the students will spend, you know, pretty much Monday through Thursday, every afternoon, in some cases well into the evening, using the microscopes and then using keys. So you go out in the field, you collect mushrooms, you bring them back, and you key them out using micromorphological characters, in addition to macromorphological characters, okay? One of the first things that students really struggle with is how do you go from having a fresh mushroom in your hand to creating and preparing a microscope slide that you can view on the microscope. And so what I thought would be a fun exercise would be to essentially give you a little walkthrough on how we do this. And I'm gonna switch cameras really quick here. I'm gonna go over to my GoPro camera. All right, and so can everyone see that? I gotta get some nods out there if you can see it. So what you should be seeing is my fingers, and there's a microscope slide here and everything. So what I want to do is sort of walk you through what the basic tool set is for using a microscope for doing mushroom identification. Tell you a little bit about the tools that are required for this, where you can find them, and then I'll even go so far as to actually make a slide so that you folks can see exactly how you go about doing this. Okay. So first off, I'll tell you about what we're looking at here, just the basic tools that one will need for making microscope slides to look at mushrooms. So the obvious thing, first off, you're going to need microscope slides, right? So these are, you know, nowadays you can buy everything on the internet. So, you know, companies like Amazon or some of the other supply houses, you can go on and you can buy these things. You can find them very cheaply. So you'll need microscope slides, of course. And then with your microscope slides, you'll need cover slips. Cover slips are little tiny pieces of glass. You can even see that. And those are going to go on top of the mushroom tissue that you are mounting on a microscope slide. The other really important tool is a set of fine tip forceps. So these are the kind that you can really stab your finger with really well. A lot of people use them for jewelry setting and things like that. Again, nowadays you can buy them online really cheaply, about $5 a pair. These, if you drop them once, they're pretty much destroyed if you land on the tips because they're very, very thin metal, very easily bendable, but really key for making slides. The one that always freaks my students out, double edge razor blades, right? So incredibly sharp and incredibly thin. If you're going to be sectioning mushrooms, these are so much sharper and so much thinner than a regular single edge razor blade that they do a much better job. Okay, so I've only cut myself, 20 or 30 times in the last 20 or 30 years. But, you know, you need to be careful with them, but they're incredibly useful. So again, you can buy those at drugstores, online, et cetera, et cetera. And then, of course, we need reagents. So whenever we're making a microscope slide, we have to take that little piece of fungal tissue and we have to mount it in some kind of solution. So the most basic one, of course, would just be water. So here I've just got a dropper bottle filled with water. The other one that we like to use for rehydrating material that's been dried out is 3% potassium hydroxide, or KOH as we like to call it. Again, you can just buy this stuff online these days, and you can get a jar of KOH and just make a 3% solution by mixing it with water. So easy to do at home. And then we have a couple of stains, and these are a little bit harder to find. The first one, which is a really important one, is called Melzer's reagent. And what Melzer's reagent is, it's chloral hydrate, which is a clearing agent. And what that does is it removes any pigmentation from cells. And then there's an iodine-based stain in there, which will stain polysaccharides, or sugars, that is, in the cell walls of the fungal tissue. Now, Melzer's reagent, because it contains chloral hydrate, that's a controlled substance. Okay, so you have to have a DEA license in order to get chloral hydrate. So this isn't one that you can easily get. The best way to get Melzer's reagent is to give a bottle of wine to a mycologist and trade him or her for some Melzer's reagent. Okay, better be a good bottle of wine. But no, all you have to do is ask. So, you know, we all make this. I trade with my colleagues at other universities that don't have a high enough DEA, you know, class license in their chemistry department to get chloral hydrate. If you come up and take the class with me, I always send you home with a little dropper bottle of this if you need some. Okay, or you can always email me and I can help you secure some Melzer's reagent. All right, and I'll show you how to use that one in just a sec. And then the final one, these are sort of just the most basic things you'll need. This is Congo red. This isn't necessary, but it's one of my favorite stains because it stains fungal tissue a really dark red color. And then when you mount that fungal tissue in water or KOH, which is hyaline, you get a really nice contrasty image, and it makes it very, very easy to see the cell walls of spores and other cells. Okay, so another favorite. This is one, again, if you'd like some Congo red, just ask me. I have enough to make about 100 gallons of this stuff to last, you know, many, many mycological lifetimes. So it's pretty cheap stuff. I'm happy to give that away. So those are the real basic tools and reagents that you'll need to get started if you want to try doing microscopy for mushroom identification at home. Okay. Now, normally what if we were doing this up at the field campus, we were in the middle of class, we would have hundreds and hundreds of fresh mushroom specimens around us to choose from. Unfortunately, it was 108 at my house yesterday, so most of the mushrooms, if they're around, look like this. Okay, so this is actually a dried mushroom specimen that came out of a fungarium, which is the fungal version of a herbarium, for those of you that don't know that word. And what this is, it's a dried mushroom. It was collected fresh, and then I cut it in half, and then I put it on a food dehydrator. And I left it on that food dehydrator for about 24 hours until it was completely dried. And then we seal them either in plastic bags or envelopes or boxes, and we put them in cabinets. And we can store them this way for hundreds of years, if not longer. I've looked at specimens that are over 200 years old, okay? So it's a really great way to preserve things. And there's no difference between looking at a dried mushroom under the microscope versus a fresh mushroom. The only thing you have to do is rehydrate the old dried mushroom first. And the first mount, a type of mount that I teach all of my students to make is really just something where you're going to tear off a piece of that mushroom gill, and you're going to look at that under the microscope. And looking at that is going to allow us to see a number of different characters, which I'll talk about in a few minutes, and I'll show you some nice photographs of them as well. Okay, so the easiest way to do this, if you go out in the field and you collect a mushroom, you bring it back, you take your fine tip forceps like this, and you literally are just going to rip off a hunk of gill tissue. So you can see that in the camera, hopefully. Small little piece of tissue. And there's an old saying in microscopy that less is more. And what that means is that you don't want to have too much tissue. You want to get the smallest, thinnest piece of tissue that you can, and that's going to make things the easiest to see once you put it under a microscope. All right? So once we have our tissue, we get our microscope slide out. I'm just opening up the box here. If I can get slides, love to stick together. So you get out your slide, and you're going to essentially, since this is dried material, the first thing we need to do is rehydrate it. Okay, so we need to get it essentially fleshy and soft again. And today we're just going to use water. Okay, so what I'm going to do is I'm going to take my water bottle, a little dropper. I'm just going to add a couple drops of water right on top of that tissue. And then we're just going to let it sit for about 5 minutes and rehydrate. And so it's going to be just like a sponge. those cells are just going to essentially absorb that water. They're going to imbibe it, and they're just going to swell up and go back to their original shape. Now, I'm not going to make us sit here and watch this for 5 minutes. That's not very exciting. So I set one up earlier. And so here's a piece of tissue that I set up while Tanya was giving her talk. And it's been soaking for about 10 or 20 minutes now. So it's nice and hydrated. And what we want to do is get it ready to stain it. And so those of you that have spent any time in biology labs in your life will recognize these. These are essentially the biology lab version of Kleenex. They're called Kim wipes, and they're just tissues. And we use them in the laboratory for wiping up excess reagent. Okay, and you can use them for wiping down certain things. The one thing we don't do with them is clean eyepieces. So these are not something you ever want to put on fine optics because they will scratch them, but they're really great for mopping up reagent and things like that. So I know it's a little bit hard to see through the GoPro, but on the slide, I have a piece of tissue and there's a bunch of water around it. And I just took that Kim wipe and I folded it up. So I have a nice little corner on it. And I'm just going to use that to mop up the excess water that's around that tissue. And that's just a really easy way to get rid of the water. And once I've done that, I can put on my Melzer's reagent. So I've got my dropper bottle here. I've got a few drops of Melzer's I'm going to add to the top. And we are good to go. What we need to do now is add a cover slip to this. I'm going to tilt that so you can all see it a little bit better. We're just going to cover that with a cover slip, and then we're ready to put it on a microscope and start looking at it. Okay? So I'll show you how to put a cover slip on, because it's sort of can be a little bit tricky. The way I like to do it, and the way I teach my students to do it, is I like to place my cover slip at a 45-degree angle to my slide, and then just drop it into place. And what that's going to do is going to help minimize air bubbles from getting underneath the cover slip. So I'm just going to, you can see through my fat fingers here, I've got my cover slip sort of at a 45-degree angle. I'm just holding it in place with my forceps, and I'm just going to drop it down right on top of that tissue. And then I'm going to take my forceps and just apply a little bit of pressure just to make sure the solution goes all the way to the edge of the glass of the cover slip. And what you end up with, if you can see all that, and put something white behind it and make it easier, is I now have my slide here, and I've got a piece of fungal tissue, it's a piece of the gill, and I've stained it with Melzer's reagent. I've added a cover slip over the top of it, and then I've squashed it down. Okay, so this is now a slide that's ready to be put on a microscope and viewed. And I do not have the ability to show you in sort of real time what this looks like on a microscope. We couldn't get the camera to talk to the PC version of Zoom off of my microscope, but what I do have is a bunch of lovely slides. So I'm gonna leave the webcam now and go back to my regular camera. Hi there. And then I will show you, go back to sharing my screen. It's a PowerPoint. All right. And so now we're back, and we can talk more about these micromorphological features. So let's just say you've gone out, you've collected your mushrooms, you've brought them back, you've torn off a hunk of a gill, put it on a slide, added some Melzer's reagent to it, put a cover slip on top of that, and you've put that onto the microscope, and you're ready to start viewing it. And if you're a brand new student and you do this, you're going to look at this and say, I have no idea what I'm looking at. Is this going to be a big slab of tissue? It'll probably be spores floating all over the place. And so part of learning to do microscopy for mushroom identification is learning a lot of the terminology that goes along with it, okay? And learning sort of where to look for certain characteristics on a mushroom. And so this is just a basic slide that I use in my courses. And so here we have just your basic mushroom. We've got the pileus or the cap, the area where the gills are located. In this case, it's called a hymenophore. And that's really the sexually reproductive area of the mushroom, okay? That's where the spores are being produced. The spores are the reproductive propagules of this type of fungus. Okay, so that's where sex is occurring on this organism. That's where it's making its spores. Some mushrooms have what's called an annulus or a partial veil, and this is just a tissue that goes from the stipe. Some people call it a stem. I tell my students plants have stems, mushrooms and fungi have stipes. So this tissue would have been connected from the stipe to the edge of the cap. And then once that cap expands and grows, that tissue sort of tears away, and it oftentimes leaves this little skirt hanging off of the stipe. And we call that an annulus. Okay, now what that tissue is doing it was protecting those young lamellae as they were developing. So it's a way to protect that tissue in the young stage of the fungus. Down at the bottom, in this case, we have a little cup-shaped structure that's called a vulva. That was actually part of another layer of tissue that enveloped the entire mushroom primordium. So when this was just a little tiny button mushroom, this species would have had this layer of tissue that enveloped the whole thing. Then as it grows and the stipe elongates and the cap expands, that tissue tears. And in some species, in some genera, you get the remnants of that universal veil as a cup-like structure at the bottom of the stipe, and we call that a vulva. And then down here we have the mycelium. And the mycelium is really the business end of the fungus. The analogy that we use in class is that you can think of mushrooms and mycelium sort of like an apple and an apple tree. The mushroom would be analogous to the apple, and the mycelium would be analogous to the tree, right? So if you think about that, an apple is just the reproductive structure. It's really the result of sex on that apple tree, right? It bears the seeds. Same thing. This mushroom, which is ephemeral and only lasts for a couple of days, is going to be produced so that the mushroom can undergo sexual reproduction, and then it's going to wither away and rot and disappear. But the mycelium will always be there. Okay, so if this is a mushroom growing in the soil, that mycelium is going to be spread as a huge network throughout the soil, and it's going to be out there doing things like breaking down dead organic matter. If it's a mutualistic species, maybe it associates with the roots of plants. If it's a parasite or pathogen, maybe it's attacking a plant host or something like that. Okay, but this is where most of the ecological interactions of that fungus occur. So for us, learning to use micromorphology, we have to understand what each of these tissues contains on a microscopic level. So just like any scientific field, we have a huge amount of jargon that goes with this. And this is probably the most frustrating thing for the majority of my students. So I'm not going to stress that at all today. It's just one of those things that you'll learn the language as you do it, you know, so as you learn to essentially more and more, you know, try and key out mushrooms or try and make microscopic slides of mushrooms and look for different cells and tissue types, you'll just pick up this language, okay? But you need to either have a reference where you can look up where, like, so if you're in a key and it says, you know, you have to determine, say, what the pileopellis structure is. Well, you have to know what the heck a pileopellis is in the first place. And so you need to be able to go back and look at some resource and determine, oh, that's this layer of tissue on the pileus surface, et cetera, et cetera. If you come and take the class, you will learn all this information and you'll just be, it'll be rolling off the end of your tongue by the end of the week, okay? So this is, as I tell my students, there's 4 things you need to learn how to identify right off the bat if you want to use a microscope to identify mushrooms. Okay, and there are 4 different cell or tissue types. The first of them is going to be the spores. And since we're talking about mushrooms here today, those are members of the Basidiomycota, so we call their spores basidiospores. And so what you're looking at here, I know this looks probably very, very strange to you. And so I've got a little helper. This is a diagram from Michael Coe's website. So here we have a mushroom. And what Michael has done here is he's drawn this as if he can just magically take this perfectly long, thin, rectangular slice right out of the middle of the mushroom and pull it out. And then that's what you have sort of enlarged over here. So this sort of olivaceous gray layer on the top here, that is the pileopellis. And that is just the outermost layer of cells on top of that mushroom cap. So it's just sort of like we have an outermost layer of cells that make up our skin. The mushroom does too. And in many cases, that's going to be an important thing for us to look at. Underneath that, we have sort of the meaty part of that mushroom cap. We call that the context or the trema. And then what you're looking at here, this is just a thin section through the mushroom gills. So keep in mind that the gills on a mushroom radiate from the stipe to the edge of the cap like the spokes of a wheel. Okay, so what Michael has done here is he's just showing you as if you were slicing through, you know, what would be a long blade-like structure. He's just taking a thin slice through it. And so here you're looking at, you know, the tissue that makes up those gills. This would be the edge of the gill down here facing the ground. And this would be the side of the gill. You know, if you had these 2 plate-like structures right next to each other, they'd be facing one another. Okay, I know that's probably super confusing. There's just not an easy way to diagram this other than just trying to imagine that you took a thin slice right through the cap of the mushroom. Okay, so the 4 cells I tell my students that they need to learn how to find and how to interpret include the basidiospores. And so what you're looking at here, this is the fertile portion of this mushroom. There's all these cells here and those are called basidia. Basidia are where sexual reproduction takes place, and that's where the basidiospores are formed. And so in this little diagram here, you see a little basidium, this little cell, and you see these 1, 2, 3, 4 little tiny small spores coming off of that. So you need to learn how to recognize what is a basidiospore and not an air bubble, okay? And then you need to do things like determine the shape and the size and any chemical reactions of those spores. And I'll show you photographs of those in just a minute. The other important cells are these sterile cells we call cystidia. So on the very edge of the gills in some species, they have cystidia that we call chylоcystidia. And then on the sides of the gills in some species, they have cystidia that we call pleurocystidia. So students, and these, turns out these are incredibly taxonomically informative. So just like spurs on, you know, a delphinium or an aquilegia or something like that, the cystidia on certain species of mushrooms are really, really indicative of a genus or a species. So really important to learn where to look for them and how to interpret them. Okay, so far we've got 2 different types of cystidia, we've got basidiospores, and the last tissue I tell my students they really need to learn how to observe is the pileapellis. And again, this is just that outermost layer of tissue on the cap of the mushroom, okay? So those are the 4 things you would start with. So not so bad, right? You know, you learn those 4 things, learn where they're located, learn where to look for them, and you're in really good shape for using a microscope to aid in mushroom identification. And so now we can finally look at some nice pretty pictures. Okay, so I want to show you examples of each of these different cell and tissue types so you get an idea of how incredible these mushrooms are under the microscope. Okay, so here's just a sampling of some of the more interesting basidiospores that we have. The first one up in the upper left here in blue, this is a species of Entoloma, and the genus Entoloma is characterized by having spores that have angled walls. Okay, so just like Tanya was talking about, where looking at the leaf types and things like that in Ranunculaceae, you can do that with certain genera of fungi. So if you get a mushroom, you make a mount of a piece of the gill tissue and you look at it and it has these angular spores, chances are you have a species of Entoloma, okay? Inocybe, so this is one of my favorites, this is Inocybe asterospora, really great name for a fungus based on its spores. So if you look at these, they're these asteroid or star-shaped spores, and these are just these big, thick spines that are coming off of the cell walls of those spores, which is really cool. Down here we have a species of Lactarius, and what you're looking at is these are the spores themselves, and then they have these reticulate ornamentation all over the surface, and that reticulation is actually raised off of the surface of the spore, so it's standing up in the air, and they form these really distinctive reticulate patterns all over the surface. In this case, those spores are staining blue in Melzer's reagent, okay, and that's an important feature which I'll talk about in just a minute. And then finally over here we have a species of Gaudieria. This is one that we get up at the field campus every year, and this fungus has these sort of longitudinal ridges on its spores. So it almost looks like little melons or something. Okay, so really, really pretty, beautiful, amazing cells underneath the microscope. And then, as I mentioned before, Melzer's reagent is it's a really important stain for us when working on mushrooms because it's a histological stain. And what it does is it stains cells and tissues either blue or reddish based on the type of sugars that are present in the cell walls. So if you have amylose in the cell walls, those cell walls stain blue. Okay, and so what you're looking at here in this fungal spore, there's all these little blue spines all over the surface of that spore. Well, those spines are staining blue, and we call that an amyloid reaction. Okay, and again, that's just due to the presence of amylose sugars in the cell wall. And so the upper picture here, these are smooth spores, but their walls are also amyloid. And when you see them in a big sort of mass like this, they sort of have that bluish gray cast to them. Okay, and so that can be a really informative character that you're almost always going to have to determine working with certain groups of mushroom forming fungi. The other character I have right next door is called dextrinoid. And so if there's dextrose sugars in the cell walls, that Melzer's reagent causes a red or a reddish brown reaction. Okay, so here we see these smooth spores that are stained red in Melzer's reagent. That's a dextrinoid reaction. And again, the same thing up here. Here's these ornamented spores that are staining reddish, and you can see all the tissue behind them is this yellowish in Melzer's reagent. Okay, so those are amyloid and dextrinoid reactions. And in this case, anything that doesn't turn blue or doesn't turn red would be considered inamyloid or non-dextrinoid in Melzer's. Okay, so this is really the only commonly used histological stain for most groups of mushrooms. Okay, so it's a great place to start. Usually if you're using keys to determine what a mushroom is, one of the first things you have to determine is whether or not the spores are amyloid or dextrinoid or neither. Okay, so it's not too hard to do, it's pretty easy. All right, so let's go on to the pileopellis now. So keep in mind, remember the pileopellis is just this outermost layer of cells on the cap of the mushroom. And we have lots of different types. Okay, so what I like to think about when I look at these under the microscope is, what does this make the mushroom look like macroscopically? So in this case, this is called a trichoderm, and what you're seeing is all these little upright hairs sticking up off the surface of the mushroom. So you can imagine that if you were holding this mushroom in your hand fresh and you looked at the cap really closely, it would have little minute hairs all over the surface. Okay, so they might look like little minute pubescence all over the surface. This one right here is called an epithelium, and it's just a layer of big fat globose cells. So you can imagine that a mushroom like this, if you looked at it, would probably be really smooth. It wouldn't have, no hairs or anything like that. This one down here, this is a layer of what's called broom cells. And they're called broom cells because they reminded early investigators of brooms. What you have is sort of a fat little cell with little finger-like appendages sticking up in the air, and they tend to have pretty thick walls. And so you can imagine that if you looked at this mushroom cap very closely, it must look like it's got this little tiny hairs all over the surface, and it does. In this case, this one actually looks like it's got suede or velvet. And it's like this, almost like this velvety-like appearance. And if you look at it with a hand lens, you can see it's all this like thousands and millions of these little tiny minute hairs that give it this really amazing appearance. And then finally, the last one is the most boring. This is called a cutis. And what a cutis is, it's just all these long filamentous cells just kind of interwoven with one another and laying on top of each other. And again, a mushroom with a cutis is typically going to have a very smooth cap, or maybe a cap that looks like it's got oppressed fibrils on it or something like that. Okay, so those are just 4 different types of pileopellis structures or types that you would have to be able to, look at and determine under a microscope. And if you come up and take the class, I'll show you all sorts of different ways that you can section a mushroom to see these types of tissues easier. Okay, it's part of the one slide that I made as an example for you. That's just one type of mount, but there's about 10 different mounts that you can make so you can see different types of characters. All right. And then finally, I saved the best for last. My favorite cells of all are the cystidia. And again, cystidia are they're sterile cells, so they don't produce basidiospores or anything like that. Frankly, we don't know why they're there in most cases, but they're incredibly beautiful under the microscope, and they're very taxonomically informative. Okay, and so this is just a slide from an old monograph here, just to give you an idea of how variable in size and shape and ornamentation these types of cells can be. And again, we have 4 different types of cystidia that occur in mushroom-forming fungi, and those are all essentially named on where they occur. So I've already showed you 2 of those that occur on the gills. We have Cheilocystidia and Pleurocystidia that occur on the gills. We have certain types that occur on the pileus. Those are called Pileocystidia. And then we have another type that occurs on the stipe surface, and we call those Caulocystidia. And again, these are really taxonomically informative. So for certain groups, you have to look at the different types of cystidia located on all 4 of those possible locations in order to really put a name on them. Okay, and so let's look at what some of these are. They're just incredible under the microscope. So this is one. This was a mount that's pretty much just been squashed out. So this is a cystidium that popped off of the gill, and it's just floating free in the mount. But just look at these things. It's just this crazy clavate cell, and then it has these weird little tapered things at the end. This is this is on a gill of a species of Conocobi, and it's just like this fat little bowling pin with a little round head and then a fat little base down at the bottom. These are some from the genus Inocobi. And so what you're looking at here, just so you can orient yourself, these are basidia here. Remember, basidia are the spores where sexual reproduction happens. And then these are the basidiospores. My mouse will come back, there it is. These dark things that you see in the slide, those are basidiospores being produced by basidia. And then this is a cystidium here. And up at the very top of it, it has crystals. So these calcium oxalate crystals are formed at the tips of cystidia in some genera. Now, as I said before, we really don't know what the function of most cystidia is, but this is a case where I would venture to guess that those calcium oxalate crystals may be there to deter little insects or other arthropods from munching away on the gills of this fungus. So keep in mind, that's where the fungus is making its reproductive propagules. So it probably will do things to deter insects and other arthropods from eating those tissues. And if you've ever tasted calcium oxalate by eating oxalis or any of those types of plants, it's not very pleasant, right? So this could be a way that this fungus is deterring fungivory, so deterring things from eating it. Not tested, but hypothesized. All right, and then here's my personal favorite. I work on the genus Mycena. It is a beautiful genus, both macroscopically and under the microscope. It perhaps is even more beautiful. You get these incredible cell types, different types of cystidia on all the different surfaces. These are cystidia from the gills. And so this one here, they're like these big inflated cells, and the entire surface of them is just covered with all these little finger-like projections that are sticking off. This one up here is sort of the same thing. It's a clavate or club-shaped cell that has all these little tiny spines all over the surface. And then the very tip of it is elongated into this incredibly long, sort of tapered flexus little excrescence. Very bizarre. And then this one down here is just craziness. This reminds me of sort of balloon animals gone bad. It's just this very strange looking cells that, what is the function of these? We really don't know. What's amazing to me is that they are evolutionarily and genetically conserved. So what we see is that these things, as crazy looking as they might be, are consistent from one species, or within that species, from one collection to the next. We will see these things being identical in size and shape and ornamentation. So pretty wild, all right? And just a few more, because I think they're beautiful. This is another species. These are Chylocystidia. So this is the edge of the gill right here. And that whole edge is just covered with these cells of these, these long pointed tips to them. This is mounted in Melzer's reagent, so it has that sort of yellowish brown color to it. And then this is an example down here of that Congo red stain. So you can see how it's picked up that red stain really well. And you can see those cells really easily. I've kind of squashed that one out a little bit, okay? And then keep in mind there's other types. So I mentioned that on the stipe surface, we have Caulocystidia. And so here we have, these are Caulocystidia on a species of Mycena. And these are just these long filamentous cells sticking out, and they're just covered entirely with these little spines. And then one of my personal favorites, this is the Caulocystidia on a species of Hemimycena. It's called Hemimycena tortuosa. And if you can see these, if I get my mouse to work, if you look at this one up here, these things are shaped like corkscrews. And at the very tip of them, they have a little globular head that exudes a sticky droplet. So these things come off the stipe and they grow in a corkscrew fashion. And so you get, under the microscope, you just see this whole surface with all these crazy corkscrews sticking out of it. And then each one of them has this little sticky drop at the end. Just totally bizarre, right? So that's Hemimycena tortuosus. Those are considered a tortuous cystidia. And then finally, this is Pileocystidia. So this is, again, this is the cap of the mushroom, what you're looking at here. I've sliced through the mushroom really thinly. This is a species that we get up in the Sierra. These are the gills down here. And then this is the mushroom cap up here. And the whole surface is covered with these sort of bowling pin shaped cystidia. Those are called Pileocystidia. So again, I know lots of terminology. It's just something that you'll learn by doing. And if you come up and take the class, I can guarantee you'll get to see all these things. And, as I said, you'll learn this jargon very quickly. Okay. So hopefully I've convinced you that using a microscope to view mushrooms is a good endeavor, something you definitely want to do. And I really do hope you'll come up and take the class. I think you'll really enjoy it. And I think that's all I have for you today. Okay. And I'll let you, go back to Mary. Stop sharing my screen.
Thank you, Brian. Oh, the enthusiasm of Brian and Tanya are just infectious. And I know we've all been kind of starved for our week of learning, or at least our 1 week of learning at the field campus. And I'm going to put my hat on now as the treasurer of the Sierra Nevada Field, the Friends of the Sierra Nevada Field Campus. We've done a lot of improvements on the campus so far, and our next big push is to remodel that lab that you saw pictures of in Brian's slides. His class is at the end of May, and it's often snow on the ground. It's freezing outside, and it's really cold in that lab, right, Brian?
Yes, very cold. So I have to say thank you to the friends for all the work that you've done up there. Fantastic improvements. Will make teaching there so much more enjoyable.
Yeah, thanks. So this is the third Zoom of the summer, and our first Zoom, we had a wonderful student donor come forward and put $10,000 on our virtual table as a match. So we're pretty close to meeting that match, but I just want to ask everybody to donate often, again, more. It really makes a difference when we all pool a little bit of money together. We could make the remodel of the lab happen. And I know myself, being during the pandemic, I'm not spending as much money as I usually do. So one idea for you is to donate to the Friends of the Sierra Nevada Field Campus. I put the information in the chat. I can put it in there again. And that's all. I just want to thank you so much for giving your time. Brian and Tanya have presented 2 times in this series, and all the other instructors. And so JR is out there. Hopefully he has enough connectivity. He texted me and said he was kind of having problems, but JR, if you start talking, maybe we'll find
I'll keep my fingers crossed, Mary. Thank you. All right. Well, thank you all so much for attending this Zoom session. As Mary said, this is our third event of the summer. And I want to really give a big shout out to Mary for putting these on and for organizing them and getting them all set up. And you did a great job, Mary. Thank you so much.
Thank you, JR.
I also want to thank the Friends of the Sierra Nevada Field. You're welcome. The Friends of the Sierra Nevada Field Campus in general for raising, putting the efforts into raising so much great money that have been used for the renovation of the bathrooms and to put a new roof on the staff cabin, and hopefully soon to have the classroom renovated. Really, really helpful, and it makes us look good in the eyes of the powers that be at the university corporation, and at the university in general. And I also want to thank all of you for attending today. That really makes us feel good that so many people are still interested, even though you can't come here and enjoy the beautiful place. And so thanks to you all, and fingers crossed, we'll see you next year so I can give you all big hugs.
Feel free to unmute yourselves if you want to chat with anybody you see here that you're friends with. Like, I want to say hi to Janet because I haven't seen you for a few years. You're muted, so yeah. Thanks, JR.
It's so great to see you doing this, because I remember we talked about the need for a Friends of the Sierra Nevada Field Campus organization.
Probably 5 years ago.
Yes, it's at least 5 years ago. And you pursued it. I'm just so impressed. That's just wonderful.
Thank you, Janet.
I want to thank Brian and Tanya. Those were both amazing presentations, and I would really love to take both of your classes. It was
Thank you.
really exciting.
Good. It's really great to see everybody. See, people are starting to go away. We had, I noticed that we had over 150 signed up once we got started, but 60 came into the room. So the YouTube video will be available. If things went a little fast for you and you want to review it again, just email us and I'll send you the link. I'll have it up on YouTube by tomorrow.
Yeah, just a big shout out there for Mary. I really appreciate you keeping the spirit of the field campus alive this summer through all of this sense of community feels very involved. And I know it wasn't an easy task, so it's been really really wonderful to work with you on this. So, yeah, thanks for all your work.
Yeah.
Feel
thanks,
it.
Mary.
Feel it.
You know, it's like we're missing the place and we're missing the food, but I think we're really missing the learning too, you know, because it's just this whole package. I don't know anywhere else in the country where this is happening, you know, for a whole week, where you can take a course, be fed this incredible meals, learn, and I think if there are people here who have not been to the field campus, what's really cool is that you might be taking the fungi class, but there's a birdsong identification class going on at the same time, and you're going to be able to meet with those people, and you're going to hear some of those lectures if you feel like doing those. And so you really get more than the class that you're taking. So, yeah.
Agree. I
Yeah.
agree.
And JR, it's nice to see the sun out behind you. After all the storming, you're muted. Yeah, he's on kind of a delay because of, well, you all know what the internet's like up there.
Yeah, it's awkward for me to talk.
Yeah, but thanks for being there. It was nice to see Annie too. Yeah. We're getting lots of comments here in the chat if everyone wants to see those. Lots of people thanking us. Can't wait to return. Can't wait to come for the first time. Hoping for next year. Well done. Yeah. Is Bill Wilson, are you still with us?
Yep, he's there.
Is he? Oh, could you unmute yourself, Bill?
There I am. Okay. Had to get to the button.
We need your camera too.
Oh, okay.
Bill's the president of the Friends of the Sierra Nevada Field Campus, has been our guiding light for the last 3 years. Hi, Bill. Hi, Diane.
Hi.
Yeah, we've been, I was up there this last week, and the cabin, the staff cabin's getting some more paint. Hopefully in the next couple of weeks before winter hits, we'll have that finished. There's a lot of things going on. There's a lot of progress in the works right now. We're trying to get permits pulled through both of the Tahoe National Forest and the county. And then there's going to be some major things done with the dining hall. Don't be surprised if you see the outside of the dining hall looking a little different because the university took a good look last fall at the buildings, and surprised, they discovered that 70-year-old buildings have a few problems. And we wanted, they have been good enough, as Mary told you earlier, to put some money into the repair and upkeep that has been long overdue on some of those issues. And of course, putting some of our remodels together with those structural repairs. For some reason, people that live in San Francisco are a little bit more paranoid about earthquakes than we are at the field campus. And that's a kind of a chuckle, because up there we've got other Mother Nature issues that we're dealing with. It's just funny. The priorities are different, but we all agree that the place has always needed a little bit more tender, loving care. And thank goodness we've got the participants of the field campus now reaching into their pockets to bring a lot of that change. And we hope that when you show up, you will see a lot of pleasant changes come next spring.
Can I add on to that, Bill, just to reassure people that we are not going to change the rustic character of the field campus at all. We're preserving that, but you know, those posts in front of the dining hall are being eaten alive, and they were replaced, what, like 10 or 12 years ago. So those will have to change because we just can't afford to keep replacing them. But the look and the feel of the place is not going to change. We're just upgrading what's there.
Yeah, yeah. And of course, the buttes are not changing. Sierra Valley's not changing. And all the area in between, which we love to use as our classroom, are not changing. Marble Hot Springs Bridge is still there. All the glorious birds that are found in and around that area is just, as all of you who have been there know, it's just a huge, wonderful classroom that we enjoy with any one of our classes to utilize, so that we have can come back and discover something new each year. It's a great place.
I've been posting my pictures because I was up there, you know, last week and happened upon a huge meadow of Anderson's thistle. And just serendipitously was the migration of the rufous hummingbirds. So I went down in there in the middle. There were literally dozens of hummingbirds flying all around me. And it was just an amazing experience. You know, I didn't really expect to find too much in bloom this time of year, but it was truly spectacular. So I just encourage you to join the Friends of the Field Campus group Facebook page, because you can post there as well your pictures or your comments about the classes or whatever. And it's a good place to get some information about things that are coming up too, like scholarships and announcements.
A question for JR. After last night's storms, are there fires going on locally?
Well, there's one big fire going on over between Loyalton and Hallelujah Junction right now. So fortunately, it's going away from the town of Loyalton, and so there are very few structures that are affected. But Chilkoot and Vinton have been, and Long Valley have all been evacuated. That's the closest, and we haven't had any effect here. The wind is blowing away from us, so so no smoke, but 395's closed, Highway 70's closed.
And there are updates on the group page if you want to check that. Jim Kelly, who lives in Loyalton, he was a former director of the field campus. He's been sending some updates to us, and I'm posting them there.
Great.
Well, shall we say goodbye? Hoping for next year. Please, please, please. World, let us have our field campus next summer. Yeah.
Thank you, Tanya. Thank you, Brian.
Pleasure.
Thanks to everybody.
All right.
Thanks, Tanya and Brian. Really great job.
Thanks, JR.
Thank you, guys.
Bye.
Bye,
Okay,
everybody.
bye-bye.
Back to painting.